I'm going to go through a typical osmosis question. The structuring of an osmosis question will cover method, results, graph, and then explanation. And there's always the same questions asked each time. So with any method in a required practical or a practical question, I always check off my independent, dependent control and identify if they've got any reliability specified in the method. So as you can see, I've gone through identifying where my dependent is, controls are, and my independent variable. With the osmosis practical, you will have to do a bit of maths, a bit of calculation. You must remember this method here, this equation, to work out your percentage change in mass. As you can see, you've got your start mass, your final mass, and they've worked out here your change of mass and then your percentage. You will always get positives and then you'll start getting negatives. This is all normal. They'll ask you if one is missing to calculate the value. If you are stuck and you can't remember this, you have 4 sets of data here that you can figure out how they got from 0.21 to 16.2%. Okay, use this, maybe backtrack and see if you can work out how they've done it with, if you can't remember and you think it's maybe the initial minus final over initial, and you don't get that 16.2, then you know it's around the other way. So use what you have in front of you. This is a typical question. They will always ask this as well. Why do we calculate the percentage change? It's because our initial masses, as you can see on here, all the initial masses, even though they're the same shape, they are different masses. So therefore, to compare them, we have to calculate their percentage change because they have different masses at the beginning, even though they've been cut in the same way. Then it will go on to a graph question. You will have to plot your graphs. Remember, for independent and dependent, your independent variable goes on your X, your dependent goes on your Y. And the typical mark scheme for a graph, if it's 4 marks, which this one is, you will get 2 marks for your plotting. Make sure you have a sharp pencil and a ruler. Accuracy is everything. These are 2 easy marks to get if you have a sharp pencil and a ruler and do not miss the lines and are really accurate with your little cross, which I'll demonstrate in a minute. Line of best fit. If you've got a line of best fit and you've got a set date like that, it's a nice line through those. If they're scattered a little bit, make sure half and half each side. There are cases when it's like curved. That's normally rates of reaction or enzymes. So draw a nice straight line with a pencil. Do not use a biro. And this is not an art lesson. If you feather or you have to, you've had to redo your line again, make sure you rubbed it out. That's why the pencils is essential. And do a nice clear line like this. So this is wrong, this is right. Okay, and then when you've done your graph, go back, check your points. This is what the examiner wants. You'll get 1 mark for the labelling and the scale, 2 for the plots, and 1 for the best fit. So I have done a very rough line here. I haven't plotted it exactly, but this here's an example. If you are more than 0.5 of a square out, you will not get the mark. So if you do that twice, you've lost both marks. So I think 4 very easy marks to get there, and knowing how those, where you get those marks is also very essential. What they will then ask typically is 2 things. 1, explain, they might say why mass increases or why mass decreases, but here, why they are both positive and negative. So you must remember this is all about osmosis. Okay, we're using osmosis to find out the concentration of the water in a plant cell. So when it increases, it means that water's moving in, so it's going from a high to a low concentration, and the cell is getting bigger. When it's negative, it means the cell's getting smaller, and the water's moving out through osmosis from a high to a low concentration. So our Solution is water potential is lower for when it's negative and higher when it's positive. So the second part is use a graph to find the concentration of the solution inside the potato cell. That is this point here, okay? Wherever your line, your lovely line of best fit with a pencil, crosses your x-axis, that is what is needed in this answer here. Why? Is because 0% change, which means there's no net movement of water going out or in, that it is moving across that partially permeable membrane in equal amounts in both directions. So it's reached equilibrium. So that is actually our concentration of the plant cell, our estimation anyway. So that will be typically asked, and that is how you would work it out. The last bit, they might do a question on sources of error. Okay, in this case here, go back to the beginning with the method if you're struggling with this and think, well, where could they go wrong? So it could be chemically, it could be the concentration of the solutions are not right. You're not drying the chips properly. It actually could be equipment, maybe your balance isn't accurate. And also, if you're leaving it for 24 hours, evaporation might occur, and that would also affect concentration of solution. Here you only need 2, so any of those 2 would answer. But that is your typical structure for an osmosis required practical exam question from AQA.